Good afternoon, everyone. I'm Julia Marie B. Gallego, and I'm here with Eliza May Barrett to report the experiment number 19, which is the digestion of carbohydrates. Carbohydrates are one of the three main classes of foods. Its main function is to provide the body with energy to support muscular work, brain activity, breathing, and other important activities. It is made up of sugars known as saccharides. Most carbohydrate foods contain many saccharides linked together, which are known as polysaccharides. Carbohydrate digestion begins in the mouth and is complete when the polysaccharides are broken down into single sugars or monosaccharides, which can be absorbed by the body. The digestion of starch occurs in the mouth and small intestine. Salivary and pancreatic alpha amylase act on alpha-1,4 glycosidic linkage of starch randomly, releasing oligosaccharides called dextrins, then to disaccharides, maltose and isomaltose, and finally to glucose. Starch is a non-reducing sugar, but the product maltose, isomaltose, and glucose are. So the chemical changes occurring during the hydrolysis of starch can be followed with the use of glucose solution and Benedict's reagent. Part 1. Digestion of starch by salivary amylase. Starch solution plus glucose solution will give a visible result blue-black color for positive and the yellow for negative. Hydrolysate with glucose solution will have a gold-colored solution for the positive and yellow-orange for the negative. Hydrolysate and Benedict's reagent will give a green, yellow, orange, or brick red color for the positive and blue for the negative. Part 2. Digestion of starch by pancreatic amylase. Test tube 1 contains 3 ml of starch solution, 3 ml of pancreatin, and 2 ml of water, while in the test tube 2, it includes 3 ml of starch solution, 3 ml of pancreatin, and 2 ml of 0.05 mol sodium bicarbonate. Shake both tubes well and immerse in the water bath maintained between 37 degrees Celsius and 40 degrees Celsius for 30 minutes. After that, we perform the Lugol's test and Benedict's test to the hydrolysate of both test tubes 1 and 2. The first and the second test tube for the Lugol's test will show color blue due to the amylose for its positive reaction and orange for the negative reaction respectively. Benedict's test, its positive reaction may vary since the presence of sugar is indicated by the development of a precipitate which may range in color, indicating the increase of amount of sugar from green through yellow and orange to red and no change in color, or blue for its negative reaction. Starches react very poorly with the Benedict's reagent due to the relatively small of reducing sugar moieties which occur only at the ends of carbohydrate chains. They do not react positive with the Benedict's test unless broken down through heating or digestion. Other carbohydrates which produce negative results include inositol. The positive reaction of starch hydrolysate with Benedict's reagent indicates that it cannot be sucrose or starch, since starch is made up entirely of glucose and aldohexose. This leaves arabinose, glucose, fructose, ribulose, and maltose. The factors needed for salivary digestion of starch are enzymes called salivary amylase, which hydrolyses starch into maltose, temperature of food, the amount of protein food needing action, the acidity of the food and/or stomach, and the presence of any digestion inhibitors such as antacids. The pancreatin contains the amylase for digestion of carbohydrates, lipase, which breaks down fats, and protease, chemicals that helps to digest food. These chemicals are normally produced by pancreas. These enzymes include trypsin and chymotrypsin to digest proteins.
Sodium bicarbonate is significant to pancreatic digestion of starch as it is secreted in the pancreas to help aid in digestion. It also helps in neutralizing the stomach acid that is generated during the digestive process and facilitates in the breakdown of certain enzymes. The factors needed for the pancreatic digestion of starch are the following enzymes. Lipase. Lipase works with bile from the liver to break down fat molecules so they can be absorbed and used by the body. Protease. Proteases break down proteins. They help keep the intestine free of parasites such as bacteria, yeast, and protozoa. Amylase. Amylase breaks down carbohydrates or starch into sugars, which are more easily absorbed by the body. This enzyme is also found in saliva. Carbohydrate enzymes break down starch into sugars. The saliva in your mouth contains amylase, which is another starch-digesting enzyme. If you chew a piece of bread for long enough, the starch it contains is digested to sugar, and it begins to taste sweet. Digestion of carbohydrates is performed by several enzymes. Starch and glycogen are broken down into glucose by amylase and maltase. Sucrose or table sugar and lactose or milk sugar are broken down by sucrase and lactase respectively. All the food you eat goes through your digestive system so it can be broken down and used by the body. Carbohydrates take a journey starting with the intake at the mouth and ending with elimination from your colon. There is a lot that happens between the point of entry and exit. The following are the pathway of digestion and absorption of carbohydrates. First, the mouth, as we begin to digest carbohydrates the minute the food hits your mouth. The saliva secreted from the salivary glands moistens food as it is chewed. Saliva releases an enzyme called amylase, which begins the breakdown process of the sugars and the carbohydrates we are eating. Second, the stomach. From there, you swallow the food that are being chewed into smaller pieces. The carbohydrates travel through your esophagus to your stomach. At this stage, the food is referred to as chyme. Your stomach makes acid to kill bacteria in the chyme before it makes it next step in the digestion journey. Third, the small intestine, pancreas, and liver. The chyme then goes from the stomach into the first part of the small intestine called the duodenum. This causes the pancreas to release pancreatic amylase. This enzyme breaks down the chyme into dextrine and maltose. From there, the wall of the small intestine begins to make lactase, sucrase, and maltase. These enzymes break down the sugar even further into monosaccharides or single sugars. These sugars are the ones that are finally absorbed into the small intestine. Once they are absorbed, they are processed even more by the liver and stored as glycogen. Other glucose is moved through the body by the bloodstream. The hormone insulin is released from the pancreas and allows the glucose to be used as energy. And last, the colon. Anything that is left over after these digestive processes goes to the colon. It is then broken down by intestinal bacteria. Fiber is contained in many carbohydrates and cannot be digested by the body. It reaches the colon and is then eliminated with your stools. And that sums up our report for today. Thank you for listening.